Hello again. Let's begin our discussion today with talking about a key element that we're going to find making up one of the basic components of most of the molecules we're going to find in living things, and that's going to be carbon. So let's take a look a little bit about the nature of carbon itself. What we're going to find is that when we look at living things and we look at the variety of molecules that they're made up of, we're going to find that carbon is one of the most common components. It's going to be one of the key building blocks of the large molecules that living things are made from. Now, one of the things we're going to do later in the semester even is we're going to ask questions about how do organisms get their carbon, where do they get it from, to build all of these various organic molecules that we're going to be seeing. So this will be an idea that we'll come back to a number of times, and so let's get a little bit of a feel for what carbon is like. First of all, when we study molecules composed of carbon, we're doing a branch of chemistry called organic chemistry. And organic compounds are molecules that can be relatively small, but some of them are enormous in size. Some of them have billions of subunits in them. And so we're going to see a wide range of these organic molecules, these organic compounds. Now, one of the reasons we can have such a variety of different kinds of organic molecules is because carbon, this atom, is so versatile. Carbon has the ability to form 4 covalent bonds, and it forms covalent bonds with a variety of other atoms. What we find is that we can take carbon and bind it to other carbons, and we're going to see that commonly in a lot of these molecules, because what we're going to find is long chains of carbons. We're going to find carbon hooked onto hydrogens as well. And as you can see from this illustration, here's a variety of ways in which you might see these carbon-containing compounds represented. Now, some of these ideas we've already talked about a little bit. Remember, you can think of molecules as having a molecular formula, that they could be represented that way. And you can see that in the second column of this illustration, something like CH4 methane. Next to it is a structural formula, but then notice that the last 2 columns are 2 additional ways we might see molecules represented. One of them is called a ball and stick model, and you can see that in this particular case, we're trying to give some shape or form to the atoms that make up the molecule, and also notice that it's giving us some information about how those atoms are aligned with each other, the angle of the bonds. Finally, the last column shows us what's called a space-filling model, and that's to give us some sense of the surface 3-dimensional shape of the molecule, how these atoms go together to give the molecule a particular shape. So take a moment to notice some of this variability. We're going to see a lot of other pictures of carbon-containing compounds, but this begins to give you an idea of some of the variations that we're going to see associated with these carbon skeletons, chains of carbons with other atoms attached to them. Now, let's go back and remind ourselves of a few features of the structure of some of the key elements that we find in living things. Notice that before we had talked about how things like hydrogen, oxygen, nitrogen, and carbon all were atoms that are in the natural condition unstable, and that they all would like to become stable. Well, one of the key things that we're going to see happening among these 4 different kinds of elements is that there are the potential to form a variety of covalent bonds. And notice that in the case of hydrogen, there's the possibility of forming 1 covalent bond. In the case of oxygen, 2 covalent bonds. If we look at nitrogen, we can see that there's 3 opportunities for covalent bonds, and in the case of carbon, 4 opportunities for covalent bonds. So you can see that carbon is the most versatile of these 4 elements because of the large number of covalent bonding opportunities that exist, because it has those 4 valence electrons. And we're going to see it bound to these other elements in a variety of different organizations or forms. Now, here's some examples of some carbon chains or carbon skeletons. You can see that one of the ways in which they're going to vary is in terms of how long is the carbon chain. And here's just 2 examples. 2-carbon chain, a 3-carbon chain. What we're also going to see is that these carbon chains can vary in terms of how the carbon chain branches. Is it a straight chain or is it a branched chain? Now, the carbon, remember we said, has the ability to form 4 covalent bonds, and so that means that there'll be the potential in some cases, as you can see in this 3rd row, for there to be double bonds between carbons. Notice the double bonding situation that exists right here. Also notice double bonding situation that exists right there. So the position, the location of double bonds in carbon chains is another way in which we're going to see variation in the carbon skeletons of molecules. And finally, one of the interesting things that these carbon chains can do is occasionally we'll find carbon chains organized into ring structures. Enormous variability, isn't it? You can imagine that if you just vary some of these features of these carbon chains, you can come up with an almost unlimited variety of different kinds of carbon skeletons. And notice in this illustration, all we're dealing with are chains made of carbon with hydrogens attached. What if I now add to this mix things like oxygen or nitrogen, some of these other elements that we find making up the molecules of living things? All right, so enormous variability. By the way, take a moment, look at this illustration, and see if you can confirm that each carbon does indeed have 4 covalent bonds. So make sure you take a look at the illustration carefully and count the covalent bonds that carbon possesses. We would expect to find that in all the different molecules that possess carbon, forming covalent bonds with the other elements. By the way, we also mentioned that hydrogen can form 1 covalent bond. So see if that's true for these illustrations. Find the hydrogens and see if they do indeed have single covalent bonds or not. Now, if I have a molecule that's composed of just carbon and hydrogen, then I can refer to that thing as a hydrocarbon. Something like the gasoline in your car is a hydrocarbon. Molecules composed of long chains of carbons with just hydrogens attached. Now, hydrocarbons come in a variety of different forms, but when we finally want to talk about the molecules we find in living things, we're going to expect to find that we'll have additional elements added to that basic hydrocarbon structure. So hydrocarbon chains are the key sort of central organizing structure of the other molecules we'll talk about later, but a very important component giving the basic structural features of those molecules. Now, one of the things we're going to see as we look at these carbon chains is that even though I might have 2 molecules that have the same molecular formula, in other words, they have the same number of atoms, the same type of atoms, we might see them arranged in slightly different ways. Now, when I have that situation where I have 2 molecules that have the same atomic composition, right, the same molecular formula, what we're going to find is that they might be arranged differently, and we call those different arrangements isomers. And we can even classify isomers into 3 basic types. We're going to see what are called structural isomers, and in a moment I'll show you an illustration of these 3 types. Geometric isomers, and then isomers that are called stereoisomers or enantiomers. All right, so now let's take a look at some of these variations in form of molecules that have basically the same composition. All right, take a look here at this first one. Here we have some structural isomers. You can see that they both have the same number of carbons and hydrogens, but in terms of how that carbon chain is branched, we now have 2 different types of structural isomers. In some cases, for a particular molecule, a particular structure, in terms of numbers of carbons and hydrogens, there may be multiple structural isomers. Secondly, we have these geometric isomers. Notice the key feature of those. They have a double bond between 2 of the carbons. And there's variation in terms of how those 2 carbons involved in the double bond are attached to other components. So, for example, notice that here we have the X represents variable components. That might be 2 oxygen atoms, for example. But in one case, they're both on the same side of the molecule. By the way, we'll use the word cis to represent that kind of arrangement when there's 2 of the same type of material attached on the same side of a double bonded pair of carbons. And then over here we have those materials attached on opposite sides of that double bonding situation. By the way, we'll be using the word trans to represent that kind of arrangement. And we'll talk more about that later. Finally, here we have those stereoisomers or enantiomers. And what we see here is that there's a central carbon that's in the midst of 4 different things attached to it. Notice that that carbon has 4 different things attached at its 4 different covalent bonds. Notice also that if you look at one side versus the other, that they're mirror images of each other. In other words, the things attached to that central carbon have a different orientation. We even refer to these as being either right-handed or left-handed, and that actually has to do with what happens to these molecules if we put them in the form of a crystal and we shoot polarized light through them. The light will actually bend either to the left or to the right, and you'll see sometimes them referred to as L forms. That's for ones that are being shifted to the left, and D forms, those that shift light to the right. So later when you hear about L isomers or D isomers, this is what it's going to be in reference to. Now you might say to yourself, well, heck, these things are all composed of the same stuff. They just have little variations in terms of how that material is attached to each other. The interesting thing is this can make a huge difference in terms of how cells make use of these particular molecules. We're going to find that in many cases cells will only use one isomer and not the other. In other cases, they might use both isomers interchangeably. In other cases, the cell might actually take one isomer and change it into the alternative form so that it can make use of it. So these isomers may be important in determining the functional qualities of a particular molecule. Make sure you take a note of this illustration and look at it carefully. Remember what I suggested before. Practice looking for the number of covalent bonds. Use this illustration to do that type of thing as well. Now, here's one example of how stereoisomers can have significantly different functions. Here we have 2 versions, L-DOPA. Remember I said that L means that if I took this molecule and formed it into a crystal and shot light through it, it would bend the light to the left. The L stands for levorotatory, which means bending to the left. And the other version of this molecule, the D-DOPA, if I took it and placed it into a crystalline form and shot light through it, it would bend light to the right. The D stands for dextrorotatory, which is another way of saying bending to the right. So, look at the difference, though, in terms of what they do. If I gave somebody L-DOPA, it would be able to attach to nerve cells in their brain and would have an impact on cell function. The D-DOPA form can't do that. So even though these 2 things are composed of exactly the same groups of atoms, because of the difference in the arrangement of those atoms, one has a particular function and the other one lacks any function. That's an important idea to keep in mind, and it's something that we'll hit over and over again this semester. How little differences in the structures of molecules, the arrangement of the atoms making up a molecule, can relate to big differences in terms of how they're used by living things, their function, the role they have. Shape is very important for determining the function of molecules. Well, let's take a look at what we just have been discussing. We're focusing on the idea that carbon, as this central organizing molecule is, central organizing atom of molecules, is very versatile. It can form 4 covalent bonds, has the ability to form covalent bonds with a variety of other elements, and that we would expect to find that as a basic component of the structure of most of the molecules we're going to be talking about, we're going to see hydrocarbon chains, or what's sometimes referred to as a hydrocarbon skeleton. Chains of carbons with hydrogens attached, and of course we're going to be interested in what else might be attached to that hydrocarbon chain. Then we talked about the idea that even though molecules might have the same composition, the same makeup in terms of atoms, they might come in slightly different forms. Remember we had structural, geometric, and stereoisomers. And those different forms, different arrangements of atoms might be extremely important in determining the function of a particular molecule. Now we're going to build on this idea in future videos, so consider this first, and then consider how we might make these molecules, the variety of molecules, even more complex by adding other elements to these hydrocarbon skeletons. I look forward to talking to you next time.